Charles Darwin said, and that has been proven again and again, that every single species in our planet is connected with a single ancestor by a tree of life, right? How much can we infer from the fossil record that for centuries was the only source of information, right? Well, much more than we might think, right? This is a copy, a replica of a Neanderthal skull, right? Neanderthals coexisted with our species for a very long time, estimated from 300,000 to 30,000 years ago, until it became extinct. And there's a lot we can infer from anatomy. A lot of things about potential physiology, potential capacities, whether or not you walk with two legs, etc., 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 right? Some about brain, right? From the cast, the endocast, you can actually gather from this. And some inferences about the mind, because we know that these species perform some kind of burials of their family members, and that tells us something about a likely emerging abstract mind, right? What else can be done? Well, one of the great advances of molecular biology in the 20th century was the possibility of looking at information at a very different level, which is the genome, right? Finding out sequences and comparing these sequences to actually redefine the tree of life under a different umbrella, right? Very good. I use the Neanderthal case because that's a spectacular example of how much science can advance against any kind of dream that we had. 20 years ago, it would have been probably insane to think that it would be possible to have the genome of Neanderthals, right? But in 2006, and then a final effort in 2010, the whole sequence of the Neanderthal genome was found out. And that opens the door to many things. We can compare the genes from this paleogenomic data with our genes and infer many things, from physiological adaptations to the likelihood that they could be infected by some given viruses, etc., etc. And some problems that have to do with diseases that we will discuss in some other lecture. But actually, I would like to address in this seminar two important things. One is more qualitative, but very important, which is what is the evidence of evolution, the actual evidence that is obtained by looking at genomes and comparing molecules, right? And not only molecules, but also other scales in the genome, right? We'll see now some examples. And a more fundamental problem, right? Which is the following. We live at time t now, right? So the real world now. I have the sequences of, for example, if I look at primates, right? I can have sequences from the genomes of chimps, bonobos, gorillas, orangutans, right? Great apes. And we know we are close, since the times of Darwin and his colleague Huxley, we are clearly related, right? For a number of traits. How do I know how far is in the past our ancestor? So at some point, something that was not a human, not a chimp, diverged and generated different species. Can we calculate that knowing only the information from today, right? We will find out how to do a mathematical model for that. What is the evidence for evolution? And I'm mentioning that because there's been, and still there's this argument that some non-physical things have acted, for example, in human evolution. Darwin was bold enough to actually say in The Origin of Species, that was not a good time for saying that also humans are under the force of natural selection, right? We are not special in that perspective. Creationists and other people pretend that we were somehow created separately from anything else, but the genome and anything we can actually compare clearly says that that's not the case. Interestingly, within the genome, we have also a fossil record, right? Now we can see in which way. For example, our genome is filled of all kinds of inserted sequences. Many of them are of viral origin. You'll find out in your genome hundreds of sequences that correspond to, for example, retroviruses, right? Many of them don't do anything. They're just there and frozen, right? And you can actually go and compare these frozen viral sequences that do nothing with the sequences that you find out in related species and find out that they also have them, right? Look clearly pointing that whatever that was before, right, was invaded by that sequence, and we just inherit that accident, right? Except that we accumulate mutations in different ways, okay? Other examples, we cannot process vitamin C, right? There are other vertebrates that can do that because they have the enzyme, right? That's actually able to, that they don't have to eat that vitamin because they can actually synthesize. We cannot. We have to take it from our diet, right? All right, but if you look at our genome, there's what's called a pseudogene. It's the same sequence that you see in those species that do have that gene that synthesizes vitamin C. We have the gene, but the gene became useless, and it's just there. It's the fossil of our previous past, right? So again, pointing to a past where we were also able to do that, right? But in our species, that's not the case anymore, right? The fossil is there. And another example that is nice to point out at a kind of larger scale is that in terms of chromosomes, right? It's large scale. So humans, we have 23 pairs of chromosomes, right? Or diploid setup, and great apes have 24, right? And so when you compare, essentially you say many things are exactly the same, but there's a number there that is curious. It was conjectured at some point, right, from several evidences, that what might have happened at some point in evolution is that two chromosomes, right, from here, got fused. Chromosome fusion is nothing strange. It happens, right? It happens often, actually. And if that was the case, it was possible to test when it was available all the information, you should observe a clear pattern, right? In chromosomes, you have a central part, right? The centromere, which is a special structure that separates two basic structures, and the part at the ends of the chromosomes, the telomeres, are highly repeated sequences, very important, as we will see, for aging and death, cell death, right? The prediction was, if there has been a fusion like this, right, joining parts, you should see in the chromosome, right, both the centromere and the telomeres have very peculiar features, right? So you should observe centromere-like structures here, right, where you should observe telomere-like, not common because they are at the extremes, in the middle. That's exactly what you see, right? There was an event in the past that fused two chromosomes. Again, a fossil record of previous events, all right? And that's something we can test again and again and again. And this, actually, the connections that we can establish between species by doing these comparisons allow us to have this picture, the tree of life. Now, that's very nice and reasonable, but what about the numbers? How do I know that there was a divergence of a common ancestor 4.4 million years that separated the branch that goes into chimps and bonobos and the branch that leads to our species? How can we calculate that, right? Well, that's a very nice example of how mathematical models can really help to do that, right, in a very elegant way. Again, as usual, we'll do some number of simplifications, but the idea is the following, right? Imagine I have a sequence, right? a sequence that represents the genome at the time of divergence, right? My genome will have some given length, right? Let's say nu, to use notation we already used, okay? This is time 0, okay? And now, imagine that I have a number of sites. This could be nucleotides, and I kind of made some abuse of language. It doesn't need to be a whole genome, of course. It can be, for example, one gene or actually one protein, right? Each case has some particularities, right? But it's some large sequence I can compare, right? Very good. At some point, for whatever reason, right, speciation starts to occur, right? And I have divergence. Meaning what? Meaning that this is my genome, right? And I'm going to have two different sequences. Different in what sense? That maybe here there's been a mutation, all right? Or maybe here, or maybe in both sides, both places, at some random point, right? Okay, some assumptions I'm going to make, which are important to solve the problem mathematically, as I'm going to show. Let's suppose that mutation rates are very small, right? Very, very small. That's quite reasonable. Second, that genomes or these sequences that I'm using are quite large, or large enough, right? That doesn't have to be thousands or tens of thousands, maybe 100 could be just enough, right? Okay, and this keeps going, right? So here I will have one difference and maybe another one, right? But the thing is that this time goes, right? And where it finishes now, it ends up today. All right? So here I had this change, here I had this change, maybe some other change happened here. All right? And in the end, I'm going to see, suppose, right, a number of mutations. Okay? That's the idea. All right. This is today, right? Okay, I need to figure out how to compare sequences, because in the end, you have the intuitive idea that is the following. The more time has happened to separate these two species, that the longer the divergence time, the more changes I'm going to observe, right? The idea is if mutations happen at random, right? And we have estimations of this. We have estimates of mutation rate. If mutation I can consider more or less constant over time and happens at random, the more time, the more mutations, okay? So here, right, I can make a measure, D, right, which is the number of differences between the two sequences, right? Which in a way is scales as the number of mutations that have happened in both parts, all right? So what I want to do is actually to find out a differential equation for D, how D grows in time, all right? And then use whatever I have to actually calculate if I was able to go back in time, how long is the time I have happened, right? Okay, let's do it the following way. Imagine I take D at some point in the future, meaning what? Well, at some point like here, right, between these two sequences, if I look at the fraction of size, think fraction means between 0 and 1, right? My number, what fraction of the sequence, right, has changed. That would be the E of t, right, because it sometimes have happened at any time. And imagine that from here to here, there's some time scale, right? Whatever it is, it could be a million years, for example, or whatever it is. Instead of going directly into the differential equation, right, I'm going to start with a discrete approximation which is more intuitive, right? The number of differences in the future will be the number of differences that I already have, well, I inherited from the past, right? Plus what? And here's the big thing, right? Well, clearly, the larger mutation rate, the more differences I will have. That's for sure. All right? That's a reasoning that we use in the first lecture on exponential decay or exponential growth. If I think in these intervals of time, of course, the larger the interval of time that they wait, the more mutations are accumulated. That's quite clear, right? Now the important thing, what is missing here, right? I can accumulate more mutations, right? But only if I consider what part of the genome has not yet mutated, right? So I can keep going on the fraction of my chain, right, where I haven't yet made changes, right? So that means that if D is the number, if you want, for example, bits that have been changed, 1 minus D is the fraction of bits that haven't not yet changed, all right? There's only one thing that is missing here, is that this happens in this branch independently, right? But also in this branch. If it happens here or here, and there's two possibilities, I will have increasing divergence. So there are two factors here, all right? So that would be the discrete form of my equation for how changes accumulate, assuming this, right? Large sequence, small mutation, right? Okay? I'll go into the end and then ask you about how to make the more exact model, right? Well, this is nothing but something that we are already familiar. If you put this on the other side and divide everything by delta t, you have the rate of increase discretized, but in the limit, what I'm saying is that the speed of change in terms of divergence is 2 mu this. This is a differential equation that we need to solve, okay? We already know about these differential equations. Again, something that has re-emerged again and again, that can be easily separated, all right? And let's think about how to write down this. I need to integrate this, and what are the limits, right? The limits are, sometimes in the past, right? Let me put this here. In terms of divergence, right, when this starts, right, this was 0 because the two genomes were identical, right? Then this diverges in time. But I don't want to have an arbitrary thing, right? I want to actually have, this is, let me write down this more clearly, this is d of actual time, all right? Divergence now. And if you want, let's write this way. The time of origination, right? And the actual time, okay? I could set up this to 0. I can, but you'll see this exactly the same thing, right? What I have is, this I know. This is the logarithm of y minus d with a minus sign, right? and this is 2 mu actual time minus original time, right? But this is, in reality, is the total time, so the divergence time, how long had happened, right? This is time of divergence, right? From everything started to mutate until today, all right? So if I put this minus sign here, I'm almost there. I can actually give you the solution, all right? Because it's something that we already have done, right? Just a little algebra, and the final result is this. Okay. And is this something we can calculate, actually? And the amazing answer is yes. This is now, right? I take the sequences, look at how many differences between them, right? And this is an estimator that I have. You can estimate the actual rate of mutation, right? Of course, the accuracy of that depends on what kind of sequences I observed. There has to be some variability, but I can do estimates, right? And quite good. So it's something I can calculate. No problem with the minus sign here, because this is between 0 and 1, right? Again, this is actually the expression that allows me to say, given two species today, right? How far is in the past their common ancestor? You see, this simple mathematics allow to actually find out crucial parts of how the tree of life is structured. As an exercise for you, let's consider how you can actually improve this model, right? Taking into account, I'm sure that some of you have already thought about that I have excluded the possibility, right, that another mutation occurs in the same place, right? This is one of the things that we should care about. If the sequence length was now so large, the smaller the sequence length, the more likely is that things are repeated or replaced, going back, for example, into a previous state, right? Back mutation. So how will you improve this model, you know? And you have to think about the corrections in mutation rates so that you are more exactly approximating the problem in a more general terms, all right? But as I was saying, that's one way of showing that you can actually calculate something that seems impossible to reach, right? And again, have in mind that even if we didn't have a fossil record, there's a fossil record in the molecular biology of gene sequences that, again, shows us how you connect with the past. And as you also discuss when we think about Darwinian medicine, right? Sequences from the past and sequences that connect us with pandemic events, for example, help enormously to actually rethink the way we look at disease.